As any enzymatic reaction, it happens very fast, it is highly specific, and it gives us results quickly. Okay, unlike the uh, chemical method where boiling temperature is required, that is not applicable in enzymatic method where the reaction takes place at 37 degrees Celsius. So enzymatic method right now before beginning itself has many advantages over the chemical method. However, between the three methods of this itself, there is some advantages and disadvantages that we are going to learn in this session. So in these methods, enzymes will be used as catalyst. So based on what type of enzymes it is present, we have three different methods which are routinely used in both diagnostic and research purposes. So we have glucose oxidase peroxidase method, hexokinase method, and glucose dehydrogenase method. Okay, each method has its own principle. Some of the methods are two-step enzymatic processes, while one is has only one step. So based on this, the specificity also affects. There are certain normal constituents that are present in uh, blood and urine that is going to interfere with this. So all those details we'll see when we get into each methods. So we're going to start with glucose oxidase peroxidase method. So the principle is very simple. Glucose in the presence of water and oxygen is acted upon by glucose oxidase enzyme. This results to formation of glucose oxide. Okay, along with glucose oxide, we get hydrogen peroxide. So that is the first step in the enzymatic reaction here. Glucose in the presence of water and oxygen undergoes a reaction which is catalyzed by glucose oxidase enzyme, results in formation of glucose oxide and hydrogen peroxide. So in the second step reaction, hydrogen peroxide is split into water and nascent oxygen by the activity of enzyme peroxidase. Okay, so it breaks hydrogen peroxide into water and nascent oxygen. As you know, nascent oxygen has the tendency to oxidize certain things. As soon as nascent oxygen is formed, it will go and oxidize things. So here we are going to use a chromogen. So chromogen means, the meaning for a chromogen is color generating compound. Okay, so anything we use here is going to get oxidized with the help of nascent oxygen, and the color formation will happen. So routinely, we prefer to use either toluene or orthodianisidine, which is the one of the most common chromogens used in this method. So the color will be generated. So that color is read photometrically at 505 nanometer. So the intensity of the color generated is directly proportional to the concentration of nascent oxygen that is present, which is directly proportional to the concentration of hydrogen peroxide which is present, and that is directly proportional to the concentration of glucose. So in simple words, we can say the intensity of the color generated is directly proportional to the concentration of glucose. If glucose concentration is more, the color intensity will also be brighter. Similarly, glucose concentration, if it is less, the color generated will also be less intense. So what are the advantages of this method? Number one, as this is an enzymatic reaction, it is not going to work in absence of glucose. Okay, so this reaction is not going to continue in the absence of glucose. The whole reaction depends on glucose. That is why we get true values with this method. True glucose values are obtained using glucose oxidase method. Second thing is, again, the intensity of color is dependent on glucose. So other normal constituents of blood, such as proteins, as we discussed in the last class, it is one of the largest molecule that is present. So this molecule is not going to interfere with the test. So the whole blood does not require deproteinization. So if something does not require deproteinization, we can use whole blood for this analysis. It is sensitive. Okay, highly sensitive, is going to detect even lowest concentration of glucose, and it is applied in many automated methodologies. In auto analyzers, glucose oxidase peroxidase is one of the most common methods that are being used. Another advantage is scenes of glucose can also be measured using this method. Okay, so CSF, even though the matrix is different, we can use CSF for uh, glucose concentration in this method. However, this method has some routine disadvantages. The first disadvantage is we cannot use or apply this method on urine samples. Now, reason behind this is urine naturally contains peroxidase enzyme inhibitors. So, if we try to apply this without any peroxidase inhibitors removal mechanism, so it is going to interfere with the second step in the enzymatic process, where peroxidase enzyme is going to catalyze splitting of hydrogen peroxide. If that does not happen, there is no nascent oxygen that will be generated. That nascent oxygen is not going to oxidize the chromogen and give us the color. Okay, so that is one of the biggest disadvantages. Urine samples cannot be used in this method. So this method prevents the use of urine. Blood contains certain chemicals, okay, such as uric acid, bilirubin, glutathione, and tetracycline, which is going to interfere with the testing. Okay. So there is a normal level at which there is no interference. If you go past that level, only then these things will interfere. However, bilirubin in mainly we see in liver patients, liver disease patients, jaundice patients, bilirubin naturally is high, so it's going to interfere with the test. So that is the only two disadvantages we can make out. And one more advantage is it is very cost effective to run. It's not very expensive because it's one of the oldest enzymatic methods, and it does not require much uh, money in implementation in the laboratory. So it is one of the cheapest methods to do. The reason behind this is there are there has been advances in these methods where almost all these uh, disadvantages have been removed. However, if you see hexokinase method, it is slightly advantageous over glucose oxidase peroxidase. Okay, so hexokinase method works on the principle of this. If you can see this reaction on your screen, you see this reaction comes in glycolysis. Okay, so this glycolysis reaction is very important and it is happening throughout the body. So hexokinase is something that is already present in the cell that is going to do. So what they are saying, one of the things is it's natural compared to glucose oxidase peroxidase. Anyways, that is one of the selling points, one of the sales people tell this, but however we are going to look at this in more technical point of view. Glucose is acted upon by hexokinase and it gets converted to glucose 6 phosphate. Okay, so the phosphate group is given by ATP, which gets converted to ADP. Okay, so glucose is converted to glucose 6 phosphate by the activity of enzyme hexokinase. Okay, so this reaction requires energy. That energy is given by hydrolysis of ATP, which breaks down and it releases phosphate, which goes and binds to glucose, and ADP is generated. That's the first step. The second step in this method, where glucose 6 phosphate is acted upon by glucose 6 phosphate dehydrogenase enzyme to give us 6 phosphogluconate. Okay, so this reaction is very important here. There is hydrolysis of NADP. So interconversion happens. These both reactions are reversible reactions. NADP gives us NADPH plus H plus. Now NADPH or NADH as it is called, it's very important. This is active at ultraviolet light. So it can absorb some ultraviolet light. Okay, so concentration of NADH generated at the end of this reaction is directly proportional to concentration of glucose. As glucose concentration starts to increase, NADH concentration also increases. So it is directly proportional. However, here there is no use of chromogen unlike glucose oxidase peroxidase method. Okay, so we have to read the concentration of NADH spectrophotometrically. Okay, as it is absorbs UV light, we are going to go into UV light range for reading the concentration of NADH. So what are the advantages over other two methods that we are going to see? Number one, serum or plasma may be used. Different types of anticoagulated bloods can also be used. That's not going to interfere with the test. We can use EDTA blood, heparinized samples, oxalate or citrate can also be used along with fluoride. Okay. Urine samples can also be used because apart from the last method we had seen, there are some peroxidase inhibitors that are naturally present in urine. Okay, here that methodology is not applied, so urine is not going to interfere with this test. Any natural constituent is not going to interfere with this test, so we can use urine samples. Linearity is also quite high. Okay, so up to 500 milligram per dl, the method is very linear. So this increases the accuracy and precision of the test, making it highly accurate and precise. So that is the advantages of hexokinase. Number one, different types of samples will be used. Different types of anticoagulated samples can be used. We can use urine. This method is highly linear and very accurate and precise. However, this method has its own disadvantages. Number one, samples needs to be deproteinized. Proteins are going to interfere with the test. This is time consuming and exacting for routine use. Now, this methodology has been reduced to around 7 to 8 minutes, which is fine. Previously, it was around 15 to 20 minutes, so it takes long time. Okay, some of the methods, if you see, some of the hexokinase, cheaper version of hexokinase test takes longer time to finish. Okay, some drugs, as we are checking for patient samples more, we expect the presence of certain drugs in their system. High concentration of bilirubin, lipemic samples where lipid concentration, the cholesterol and TG concentration is more than 500 milligram per dl, may cause a positive interference with the testing. That means all these things can give us a falsely high value. Okay, so those are the disadvantages. Okay, let me again recap. Advantages: serum or plasma. Different types of anticoagulated samples can be used. Urine sample can also be used. Highly linear and highly accurate and precise. So we don't have to use different methods for different samples. That is the advantage. However, the disadvantages are also similar. First, any method that you apply manually or automated, the samples need to be deproteinized. That means additional step is added, and additional chemical is added that's going to interfere. It takes time. Any additional step you add into your testing method, it takes time. It is exacting for routine use because it's going to take longer time. Certain drugs, bilirubin, lipemia can also have a positive interference with testing. Right, going into our next method and final method is a glucose dehydrogenase method. Now, the advantage of this is it's a one-step enzymatic process, okay, where glucose is converted to gluconolactone by the activity of enzyme glucose dehydrogenase. Now, here NADH will be hydrolyzed, it will be utilized to give NADH plus H plus. Okay, glucose converted to gluconolactone by the activity of glucose dehydrogenase. NADH is utilized and we get NADH. The concentration of NADH is directly proportional to concentration of glucose. It is one of the biggest advantages that it is only one step. The less chemicals you add, less constituents you add into the reaction, the chances of interference with these substances are also reduced. Okay, because of this reason, out of all the enzymatic methods, glucose dehydrogenase method is the most specific type of method that we can use for glucose estimation. It's highly specific. Because of its high specificity, it is also highly linear. Okay, it can measure up to 1000 milligram per dl of glucose in any sample. However, the advantages ends there. It has major disadvantages. Its cost. It's very expensive to used for routine use. Okay, routinely we cannot use it because it's very expensive. And second thing, because of its expensive nature and its costly testing price, it is not available for use in analyzer. So we cannot automate this method. This method can only be done in manual. That's why the use of glucose dehydrogenase method is limited to research purposes. It is not used in routine diagnostic purpose. Okay. So with that, we are at the end of the session.